Okay, so today we're going to go through the metabolism of other fuel sources. We're going to talk about the metabolism of proteins and lipids. So what we have focused on up until this point is the metabolism of carbohydrates and primarily glucose as the carbohydrate that we've talked about. So here is glucose here. This is the reactions of glycolysis to make pyruvic acid. You know that when oxygen is available, pyruvic acid is going to get converted to acetyl-CoA, and then from there it can enter the Krebs cycle. So you have your series of reactions from the Krebs cycle that's going to further oxidize glucose. You make a little bit of ATP from glycolysis and Krebs cycle, but more importantly, you will pull off electrons from these metabolic intermediates. Those get transferred to the electron transport system to make ATP. So we'll first talk about the metabolism of proteins, and then we'll go over lipids, but notice what happens with proteins and where they enter these cycles. So proteins get broken down into amino acids. That's digestion. That has to happen before you can absorb any amino acids across the lining of your intestines. So amino acids are going to contain nitrogen. And so nitrogen waste has to be eliminated out of your body, because if nitrogen is allowed to hang out, it will get converted to ammonia and affect the pH of the cell. Some amino acids can be converted to keto acids. Pyruvic acid, for example, is a keto acid. Some of them can be converted to acetyl-CoA. The group of amino acids that can get converted to pyruvic acid, you can run these reactions in reverse if it's any cell to make fructose 6-phosphate or glucose 6-phosphate, which the cell can then use to make glycogen or can use for cellular metabolism. The group of amino acids that get converted to acetyl-CoA, there's a lot of things that can be done with acetyl-CoA, specifically the acetate. It can enter Krebs cycle. I'll show you a slide that has a lot of the different fates of acetyl-CoA. But a lot of these amino acids can enter the Krebs cycle at different points, depending on what metabolic intermediate they get converted to. Over here is the metabolism of lipids. So we'll focus on the metabolism of triglycerides. Oh, excuse me. Triglycerides are made up of 3 fatty acids and glycerol. Glycerol gets converted to 3-pgal, and it can enter glycolysis at that point and then continue to follow through the reactions of glycolysis. And when glycerol enters glycolysis as 3-pgal, you end up making 20 ATP. The fatty acids will get broken down to individual 2-carbon subunits, which are acetates, and those will then be metabolized to make ATP. The individual acetates, coenzyme A attaches to them, so for every acetate that is liberated from a triglyceride, that's going to count for 1 turn of the Krebs cycle. So your body has the ability to metabolize all of these fuel sources, and it does as long as you're eating all of those fuel sources. Some tissues are more adept at metabolizing a certain type of fuel source over others. It just depends on the metabolic activity of the tissue, how much ATP it needs, and then what fuel sources are available, what are you eating in your diet, and whether you're eating at all. Okay, so one thing you have to keep in mind when we talk about all of this is that we eat complex carbohydrates, we eat proteins, we eat lipids in our diets. Proteins have to get broken down into amino acids. Amino acids get absorbed across the lining of your intestines. Some of them will get processed by your liver. Some of them will get shipped off to cells. The cells will take amino acids, and if they need to make a protein, they will build the protein inside of the cell. So you have to keep in mind that a protein that you get in your diet doesn't get shipped off as a whole protein to a cell. It has to get broken down into amino acids first. So protein digestion begins in the stomach. The enzymes, the digestive enzymes that digest proteins are collectively called proteases. So this is a large group of enzymes that are involved in digesting proteins. The protease that's produced by your stomach, the protease that's produced by your stomach is called pepsin. And that is going to just break bonds between amino acids. There's no specificity as to which end of the amino acid it's going to break the bond. And then protein digestion is finished in the small intestine, with the proteases that are produced by your pancreas. So what are some of the things that amino acids are used for inside of cells? They can be used to make ATP. Amino acids can be used to make other things. You have two main groups of amino acids. They're called essential and non-essential amino acids. Essential amino acids you have to get from your diet. Your body has no way of making those amino acids. But your non-essential amino acids you can make from other sources. Amino acids can be used to make different metabolic intermediates, especially for the Krebs cycle. Some amino acids, if they're categorized as glucogenic, can be used to make glucose. And if this is in the liver, then the liver will run the reactions in reverse to make free glucose. If it's any other tissue, specifically like skeletal muscle, for example, and it doesn't need the glucose for metabolism, it will take that and run the reactions in reverse to either fructose 6-phosphate or glucose 6-phosphate, and then it will use that to make glycogen. Amino acids, some of them can get used to make other fats or other lipids, and then the amino acids can be used to make any proteins that the cell needs to make. So remember that your cell in the plasma membrane, there's lots of proteins. There are structural proteins, there's proteins that form ion channels, there's proteins that form receptors, there's enzymes inside of membranes. So there's a lot of things that cells need to make proteins for inside of the cell. So how do you get these amino acids? Okay, you get them from your diet, and then you also have proteins in your body. So I mentioned a second ago, depending on what you get in your diet or whether or not you're eating. If you are fasting and you're not eating, then your body will break down your own proteins and metabolize them as a fuel source. And the biggest source of protein in your body that can be broken down is the proteins that make up muscle. So those can be broken down to make amino acids. Amino acids, again, can be used to make proteins. They can be used to make other nitrogen-containing compounds. Amino acids, this right here is called transamination. So this is a process we'll go over in just a minute. It's where you transfer the amine group to something else to make another amino acid, or you can make a keto acid. So transferring the amine group from one amino acid to something else is a way to make a non-essential amino acid. Again, or you can make a keto acid. Deamination is where you remove the amine group from an amino acid, and that makes it not an amino acid anymore. That typically will make it into a keto acid. Keto acids then can be used in metabolism. They can be used to make glucose, they can be used to make fat, they can be used to run through metabolic pathways to make ATP or other non-essential amino acids. So for your non-essential amino acids, you can take certain amino acids, transfer the amine group to something else and make a keto acid, then that can be used to make different amino acids. This just goes over the 20 common amino acids that you find. You do not need to memorize the names of these or the structures or even the classification of what groups they belong to. But we do talk a lot about proteins, and if you go on to take biochemistry or a cell and molecular class, you will probably end up memorizing these. But amino acids have the same basic core structure. And then what they have, which makes them unique, is this R group. So this R group is attached to the central carbon of an amino acid. And that R group is going to be unique for each of the amino acids. And those R groups will cause that part of the amino acid to behave like other functional groups behave. So if it's a carboxylic acid, or if it's any type of other functional group, you can predict its behavior based on that. And that will also then classify it as things like acidic or basic or aromatic or allopathic, so things like that. So of the 20 common amino acids, 9 of them are essential. So these are the 9 essential amino acids. So what this means, again, if something is an essential amino acid, you have to get it from your diet. You have no ability to make these amino acids. In general, amino acids get classified as either glucogenic or ketogenic into one of these two groups. Glucogenic, ketogenic means they can kind of go in either pathway, and it just depends on what enzymes are present and what has to happen in the cell. But glucogenic and ketogenic will tell you a little bit about what happens to those amino acids and what the possibilities are, or what the options are that the cell has to use those. So a glucogenic amino acid is an amino acid that gets converted to, for example, pyruvate. An alpha keto acid, which we'll go over those classifications in just a minute. Pyruvate is an alpha keto acid. So if you can convert an amino acid into pyruvate, remember you can run the reactions of glycolysis in reverse. Pyruvate can then get used to make glucose. And again, in the liver, it will go all the way back to glucose, and then that can be moved out of the cell. It can be moved into the blood to help restore blood sugar levels, or to help supply glucose if you're actively using a lot of ATP. In other tissues, it'll take pyruvate and run the reactions in reverse to either fructose 6-phosphate or glucose 6-phosphate. And then there's lots of things that can be done from there. In the liver, the production or the catabolism of amino acids, it increases because of the need for additional fuel sources. So the two ways that you can deal with an amino acid, the way that you can convert it to either a glucogenic or a ketogenic amino acid, is either to transfer the amine group from the amino acid to something else, or to completely deaminate it or remove that amine group. So those are glucogenic amino acids. Ketogenic amino acids can get converted to acetyl-CoA. Now, if you have acetyl-CoA, you cannot go backwards from acetyl-CoA to pyruvate. So remember, once you get to pyruvate, you can run all those reactions of glycolysis in reverse. But when you go from pyruvate to acetyl-CoA, remember, pyruvate is a 3-carbon intermediate, acetate is a 2-carbon intermediate. So the reason why you can't take acetyl-CoA and make glucose is because you have to add a carbon to it, and you have to add a pair of electrons to it. There are circuits or shortcuts or detours around it where you can take some ketogenic amino acids and ultimately make one of the intermediates from glycolysis, but it requires a lot of energy to do that. So typically cells don't do that. So the ketogenic amino acids are produced by oxidative deamination. You completely remove the amine group. And so we've already talked about what can happen with acetyl-CoA. Remember, acetyl-CoA, when acetate enters the Krebs cycle, that is 1 turn of the Krebs cycle. And you know how much ATP you make from that. You know that you get 1 ATP, and then you reduce 3 NADH plus H plus and 1 FADH2. So you can determine how much ATP you get from 1 turn of the Krebs. So let me put this on here for you just so that you have these numbers. So remember, for 1 turn of the Krebs cycle, you make 1 ATP directly. You reduce 3 NADH plus H plus, and you reduce 1 FADH2. So 1 ATP is 1 ATP. Remember that for every pair of electrons that are donated to the electron transport system by NADH, you're going to make 3 ATP. So you could take this and multiply it times 3, and that would give you 9 ATP from the electrons that are transferred by NADH. And for every pair of electrons that are donated to the electron transport system by FADH2, you make 2 ATP. So you can multiply that times 2, and that would give you 2 more ATP. So for 1 turn of the Krebs cycle, you make 9, 10, 11, 12 ATP. Okay, and that's just counting the ATP that's made by substrate level phosphorylation and then the ATP that's going to be made by oxidative phosphorylation when these electrons get transferred to the electron transport system. So for every acetate that enters the Krebs cycle, you make 12 ATP. Now, when you are metabolizing fatty acids, for example, a fatty acid gets chopped up into multiple multiple acetates. And so each one of those can be run through the Krebs cycle to make ATP. So again, there's lots of things that can be used, that acetyl-CoA can be used for. It can be used to make fatty acids. It can be used to make ATP. It can be used to make other ketones, which are a type of fuel source that some tissues prefer to metabolize. But again, it can't be converted to glucose because you can't go from acetate to pyruvate, because acetate is a 2-carbon intermediate, pyruvate is a 3-carbon intermediate. So to go back to glucose, you need to have a glucogenic amino acid that gets converted to pyruvate, not a ketogenic amino acid that gets converted to acetyl-CoA. So this here shows you the groups highlighted in this dark blue or purple. These are your glucogenic amino acids, which means that they can get converted to pyruvate, and then from pyruvate, you can run the reactions in reverse to make glucose. This also shows you where they would enter the Krebs cycle if these amino acids were to enter the Krebs cycle or get converted to a metabolic intermediate of the Krebs cycle. These two groups here in green are your ketogenic amino acids. So these get converted to acetyl-CoA. And then once you have acetyl-CoA, you know that that can enter the Krebs site. The acetate will combine with oxaloacetate to make citrate, and then you just continue through the cycle. So you do not need to memorize these categories. You do not need to memorize that these are ketogenic amino acids or that these are your glucogenic amino acids. But just pay attention to these and just notice, for example, these amino acids here can enter the Krebs cycle here and get converted to succinyl-CoA. So it just depends on where they enter the cycle as to how much ATP you would make, depending on how many electron carriers are going to get reduced from the point where they enter the cycle. So it's really easy to see how amino acids can enter the Krebs cycle and be used to make ATP. And again, your ketogenic amino acids, those get converted to acetyl-CoA, and there's lots of things that can be done with acetates as well. So what do you do with this amine group? An amine group is NH2. So you want to make sure that you know what an amine group is. There are some functional groups that you need to know. We'll be talking about amine groups and a carboxyl group. This is our carboxyl group right here. It's COOH. If it's a free fatty acid or the end of an amino acid that's not bound to anything, this is typically COO with a negative charge, which is why your amino acids have polarity to them. So transamination is where you transfer an amine group from one amino acid to something else. So in this example, here's an amino acid and here's a keto acid. If you take the amine group from this amino acid and you transfer it to this keto acid, this original amino acid gets converted to this keto acid, and this keto acid gets converted to a different amino acid. So here, let me put these on here so you can follow them. So this keto acid here gets converted to, oops, I can't draw very well on these things. Let me try this again. This keto acid, once you transfer the amine group from this amino acid to this keto acid, you've now made this amino acid. So you've made a different amino acid. Again, that's transamination. You transfer the amine group from this amino acid to this keto acid, and now you've made a different amino acid. And remember, the only amino acids that can be made are your non-essential amino acids. So this amino acid here, once you remove the amine group and you transfer it to the keto acid, this amino acid gets converted to this keto acid. And then there's lots of things that can be done with keto acids as well. So transamination is where you transfer an amine group from one amino acid to a keto acid. So an example down here is if you transfer an amine group from L-alanine to alpha-ketoglutarate, so L-alanine is the amino acid. The amine group is this in blue right here. This is a carboxyl group. So this is the amine group. If you transfer the amine group from alanine to alpha-ketoglutarate, you can make pyruvate. So you have a way to make pyruvate. Alpha-ketoglutarate is a keto acid. And what you've done then is you have converted alanine to L-glutamate, which is a different amino acid. So you start with L-alanine, you transfer the amine group to make pyruvate, and then you also end up with L-glutamate. That's transamination, transferring the amine group. Oxidative deamination, to deaminate something, you remove the amine group. It's called oxidative deamination because where the amine group was originally attached to the original amino acid, that gets replaced with a double bond to oxygen. So that's why it's called oxidative deamination. Here's your amino acid. You deaminate, you remove this amine group. Oxygen comes in and binds to this to make this stable because all of your valence electrons have to be bound with something. So oxygen replaces the double bond there, and then this amine group gets released. It's going to combine with another hydrogen because nitrogen needs to form 3 bonds. So NH2 means that nitrogen only has 2 bonds. It will combine with another hydrogen to form ammonia. NH3 is ammonia. So ammonia can't be allowed to hang out inside of a cell. So it would need to get shipped off to your liver to be processed so that you can get rid of this nitrogen waste from your plasma. So this is transamination here, where you transfer the amine group from one amino acid to a keto acid. You're transferring the amine groups. You just pluck it off of this one, add it to something else. In deamination, you completely remove the amino acid. You don't transfer it to anything else. The removed amine group gives rise to ammonia. Ammonia then gets converted to urea because that's the form of nitrogenous waste that we eliminate. This is typically, oxidative deamination here is typically going to happen in your liver or your kidneys. So remember, your kidneys are involved in getting rid of metabolic waste products, and nitrogen waste is one of those things. Here, this little note here, glutamate is an amino acid, is commonly deaminated to make alpha-ketoglutarate. So alpha-ketoglutarate, you know, is an intermediate from the Krebs cycle. So there's keto acids and ketones and ketone bodies, and the terms are all very close to one another. So I just wanted to go through what the difference is with you. For something to be a keto acid, it has a carboxyl group and a ketone group. So it has both of those groups. If it has both of those functional groups, it's called a keto acid. There are 3 different alpha keto acids, and there's 1 beta keto acid. Pyruvate is probably the most common alpha keto acid, and there's lots of things that can be done with pyruvate. Oxaloacetate is also an alpha keto acid. This is the intermediate in the Krebs cycle that's going to combine with an acetate that's being donated by acetyl-CoA, and that is how you get acetate into the Krebs cycle. Alpha-ketoglutarate is another intermediate in the Krebs cycle. It also has some other functions outside of the Krebs cycle. It acts as a coenzyme in cell signaling or cell communication, and it's also involved in transamination reactions. You can transfer an amine group from something else to alpha-ketoglutarate to make a different amino acid. Acetoacetate is a ketone that is released from the liver, and it can be used by other cells that can't use fatty acids as a fuel source. So some cells, some brain cells, for example, your brain can metabolize certain types of fatty acids. They can't metabolize all of them. If it's a large or a long chain fatty acid, those can't cross the blood-brain barrier. So your medium and short chain fatty acids do, but then one thing that can happen is your liver, for example, can take something and convert it to acetoacetate that is a keto acid, and that can be used as a fuel source instead of other triglycerides. So it's just a way to make another fuel source available for some tissues that can't metabolize fatty acids. So keto acids, that's what we just talked about. So it has a carboxyl group and a keto group. Lots of your intermediates of glycolysis in the Krebs cycle are keto acids: pyruvate, alpha-ketoglutarate, succinyl-CoA, fumarate, oxaloacetate. These are all examples of keto acids. So they're very common metabolic intermediates in glycolysis and Krebs cycle. Ketones are also called ketone bodies. And there's no real rhyme or reason as to which term is used. There are 3 ketones that are produced from the liver when fatty acids are being metabolized. So we're going to go through the metabolism of a fatty acid here in a minute, and you can see how much ATP you make when you metabolize a fatty acid. So tissues that have a very high metabolic requirement, like your liver, like skeletal muscle, will metabolize fatty acids preferentially over other fuel sources because you make so much more ATP from them. So in the liver, when it metabolizes fatty acids, it can make these 3 ketones: acetoacetate, which is a beta keto acid as well, acetone, and beta hydroxybutyrate. Acetone isn't typically produced in a normal healthy individual, but if a person is diabetic and it's not being treated, this can be made, which is why their breath smells fruity. It's because the acetone, the only way to get rid of acetone is to volatilize it, so you breathe it out basically. But in a normal healthy individual, your body doesn't typically make acetone. But acetoacetate is a keto acid that can be used for a lot of different things. So when is fatty acid metabolism at its highest? When you're fasting. If you're not eating anything, your body will shift to its fuel reserves. And the energy source in our body that's a fuel reserve is adipose tissue. So adipose, you'll have lipolysis to free up the triglycerides for metabolism. So you'll get a lot of fatty acids that are metabolized. If you're eating a keto diet, right? A keto diet is very low in carbohydrates, but it's high in protein and fat. If someone has alcoholism and they have cirrhosis of the liver, then they end up metabolizing fatty acids over anything else. And again, if someone has pretty advanced type 1 diabetes. So let's talk about triglycerides as fuel. So your triglycerides, we get lipids, we eat lipids in our diet. Lipids get broken down to a couple of different forms to get absorbed across the lining of your intestine. They can get broken down into free fatty acids. They can get broken down into triglycerides. They can get broken down into a monoglyceride, which is a glycerol with 1 fatty acid attached to it. So there's a lot of different forms that they get broken down to get absorbed across the lining of your intestines. Then they get shipped off to your liver. Your liver forms different fatty acids, and then those get put back into circulation, and those can be used as fuel sources for other tissues. So any additional energy source that you've eaten that your body hasn't converted to ATP and used. So if you're, for example, eating more calories than you're burning in a day, all those extra calories will end up getting converted to triglycerides and stored in adipose tissue. So then when you need an extra fuel source, you have a reserve supply at hand. So a triglyceride is made up of 3 fatty acids bound to glycerol. Glycerol is a 3-carbon alcohol, and this is a fatty acid. So this is a common way to represent a fatty acid. If you haven't had organic chemistry yet, then what you need to know is each one of these points here, 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 all of these points represents a carbon. And these carbons are saturated, meaning all of the bonds, so you have a carbon bound to a carbon here. Carbon forms 4 bonds, so that means this carbon will have 3 hydrogens attached to it. This carbon here is bound to this one and this one, so that means it'll have 2 hydrogens attached to it. So it's only carbons and hydrogens. It's called a hydrocarbon tail. But at the end of a free fatty acid, you have this carboxyl group. So a triglyceride is 3 of these fatty acids attached to glycerol. So the way that it attaches is the carboxyl end attaches here where you have this hydroxyl group here. So that's how a fatty acid would get attached to glycerol. So this is a common triglyceride. So this fatty acid here is called palmitic acid. This fatty acid here is oleic acid. And this one here is alpha-linolenic acid. So different fatty acids have different names. And notice that they're different. This one has fewer carbons than this one. This one has a double bond here. Anytime you have a double bond between carbons in a fatty acid, that's called a unit of unsaturation. So if you think about it, they recommend in our diet that we eat polyunsaturated fats instead of saturated fats. This is a fully saturated fat, meaning all of the bonds are saturated with hydrogens. This is not a great type of fatty acid to eat because you have rotation around these bonds. And so these can pack very tightly into adipocytes, and you can pack a lot of them in there. If you have a fatty acid that has a unit of unsaturation, so this would be a monosaturated fat here, you do not have rotation around a double bond. So that means it's a kink. So if something has a double bond here, it can't pack and fold as tightly into a cell. This is a polyunsaturated fat. It has 3 units of unsaturation here. And again, this one would take up more space as well. So the reason why they recommend we eat polyunsaturated fats is so you don't end up packing so many of these fatty acids so tightly into your adipocytes. But this is what a typical triglyceride looks like. So here's your glycerol head. This was the hydroxyl group of glycerol. This is the carboxyl end of the fatty acid. So when the fatty acid is attached to glycerol, this is where that bond forms. So a typical triglyceride, you don't necessarily have 3 fatty acids that are the same. There are some of them that have them. It's called a homotriglyceride if all 3 fatty acids are the same. But it's more common to have a triglyceride where you have 3 different fatty acids. So again, your triglycerides are going to get stored in adipocytes. So this is an adipocyte right here. You looked at these under the microscope in anatomy. This is a vacuole here, which is where the triglycerides are actually stored inside of the adipocyte. Typically, this vacuole is so packed full of triglycerides that it takes up all of the room inside of the cell. So you don't even see this separately from the cell membrane. And also because it takes up so much room, it typically squishes the nucleus. So you'll see the nucleus kind of flatten on this side. So if you need a fuel source, and there's lots of different things that can induce what's called lipolysis, which is the liberation of fatty acids for metabolism. It can be induced by glucagon. Glucagon is produced when your blood sugar levels drop. So it's a way to increase blood sugar levels. So that's one of the triggers for this. So what that would mean is that if your blood sugar levels are already low, you don't want your cells to take in more glucose for metabolism. So your body would liberate fatty acids and make more of those available for metabolism so it can metabolize those instead of glucose. Norepinephrine and epinephrine, collectively you know this as adrenaline. This is released when you have sympathetic activation or your stress response. And that's something that happens commonly, happens multiple times during the day. There's lots of things that can trigger it. Some things good, some things bad. But when you have sympathetic activation, that typically is called your fight or flight response. So you're either going to stand there and fight for your life, or you're going to run for your life, which means you're going to need an extra fuel source because you're going to need a lot more ATP, especially for skeletal muscle and your liver. So this will trigger the liberation of fatty acids. Cortisol is another stress hormone that's released with sympathetic activation that causes further release of lipids, and growth hormone. If you think about it, especially when you're a little kid, then you need to have a lot of fuel sources available because you're making new body tissue. So in order to do that, you need to have a significant amount of fuel sources available to do that. So lipolysis is where the fatty acids are released from the adipocytes. So what gets released is not an intact triglyceride, but the triglycerides are broken down into glycerol and then the individual fatty acids. So again, this is going to happen in adipose tissue. So if you notice right here, here's a protein kinase. Remember, a protein kinase is going to transfer a phosphate group either to or from ATP. We'll talk about this in more detail when we go over cyclic AMP as the second messenger. But here's epinephrine binding to a receptor that is basically triggering all of these events to liberate the triglycerides. So that's called lipolysis. So exercise can induce this, fasting when you're not eating, stress and growth. And these are all hormones. Norepinephrine is also a neurotransmitter as well as a hormone, but all of these are hormones. So you have lipolysis that frees up your glycerol and your fatty acid tails. So glycerol gets moved out of the adipocyte and then it gets taken up by cells of your liver, kidney, skeletal muscle, and brain. Glycerol gets converted to 3-PGAL. In your liver, it can take the 3-PGAL and it can run the reactions in reverse from 3-PGAL to make glucose, right? That's gluconeogenesis. Or it can make fructose 6-phosphate and use that to make glycogen, which is glycogenesis. In most other cells in your body, the PGAL that is produced enters glycolysis, and what you get from metabolizing glycerol is you make 20 ATP. That's a number that you're going to need to memorize because we're going to go through and kind of add up how much ATP you make from metabolizing a triglyceride. So what happens to these fatty acids? The fatty acids are metabolized in the mitochondria of cells, and the pathway that's involved in metabolizing these fatty acids is called beta oxidation. So remember that fatty acid is a string of hydrocarbons, carbons that are saturated with hydrogens. And so what happens is those fatty acids get chopped up into little 2-carbon subunits, which is acetate. Coenzyme A attaches to it, and now you have acetyl-CoA. There's lots of things that can be done with acetyl-CoA. It just depends on what the cell needs. Right now, we're going to go through and calculate how much ATP you can get from metabolizing a fatty acid. So this is kind of an overview of the entire process of beta oxidation. So this is the catabolism of fatty acids. So the fatty acids get moved into the cell by a fatty acid transporter. It's going to bind to coenzyme A. Coenzyme A is a common enzyme that's used to help move things around and transfer them. So the fatty acid with coenzyme A gets moved into the mitochondria, and that's where you have beta oxidation that occurs. So you just have this process where these enzymes chop off these little 2-carbon subunits, and those 2-carbon subunits are acetate. Coenzyme A attaches to each of those individual acetyls, and then again, whatever happens to these acetates is going to be determined by what the cell needs. Does the cell need to make phospholipids? Does the cell need to make a different triglyceride? Does the cell need to make eicosanoids, which are chemical messengers involved in your inflammatory response? Does the cell need to make cholesterol? Cholesterol is a component of cell membranes. Cholesterol is also what all of your steroid hormones are derived from. So your sex hormones like testosterone, estrogen, progesterone, but cortisol, a stress hormone, is also derived from cholesterol. Do you need to make bile salts? These are all things. Do you need to make ATP? Right? So these are all different fates of acetyl-CoA. You can take acetyl-CoA and you can make a ketone body, or from a ketone body you can make acetyl-CoA. From acetyl-CoA you can make other fatty acids. So how do you get acetyl-CoA? you get it from glucose, because through glycolysis you make pyruvate. Pyruvate gets converted to acetyl-CoA. From your keto or your ketogenic amino acids get converted to acetyl-CoA, and then also from your fatty acids. So there's a lot of different ways that you can get acetyl-CoA, and then there's also lots of different things that the cell can do once it has acetyl-CoA. So what we're going to do is go through and try and figure out how much ATP you make from a common or average sized triglyceride. So each fatty acid will be oxidized to individual acetates, that's the 2 carbon subunits, and then combined to form acetyl-CoA. So right now we're going to assume that the cell is going to use all of these acetates to make ATP. So for every acetate that is made, that's 1 turn of the Krebs cycle. So remember, for 1 turn of the Krebs cycle, you make 1 ATP, 3 NADH plus H plus, and 1 FADH2. This here equals 12 ATP. Right, we just did that calculation. So from 1 turn of the Krebs cycle, you make 12 ATP, right? You know how to get to this number, right? Because for every pair of electrons that are donated to the electron transport system by NADH plus H plus, you make 3 ATP. So 3 NADH plus H plus times 3 is going to give you 9. For every pair of electrons that are donated to the electron transport chain by FADH2, you're going to make 2 ATP. So 2 times 1 is 2. So 9 plus 1 plus 2 gives you 12. Now the other thing is, for each bond that's broken in the fatty acid to free up those acetates, every time you break a bond, you're also going to reduce 1 NADH plus one NADH plus H plus and 1 FADH2. So for each bond that you break, you're going to make 3, 4, 5 ATP. So you make 5 ATP for each bond that's broken in the fatty acid. And then glycerol, from glycerol, remember glycerol gets converted to 3-PGAL and enters glycolysis, and from glycerol, you'll make 20 ATP. So you can go through and do the calculation to determine how much ATP you get, or you can just memorize 20 ATP. That one's a little bit easier. So this is showing you a homotriglyceride. So this is called palmitin. This is one of my favorite triglycerides because it's found in chocolate. So again, a homotriglyceride means that each of the fatty acids are identical. So here is your glycerol head, and here's your 3 fatty acids. So here's one way you can draw the fatty acid, and this way gives you an idea of what a hydrocarbon really looks like, that it's carbons that are saturated with hydrogens. Okay, this is how we typically draw it in OChem. This is another way that you would look at it, like in general chemistry when you're learning about bonds and things like that. These are all 3 the exact same thing. So basically, the process of beta oxidation is you're going to snip off this fatty acid, and then it's going to get cleaved into 2 carbon subunits. Each of those 2 carbon subunits is acetate. Coenzyme A attaches to it, and now you have acetyl-CoA, and every single one of those is 1 turn of the Krebs cycle. But also, in between the acetates is a bond that's broken. And for every bond that you break, you're going to reduce 1 NADH plus H plus and 1 FADH2. So in order to snip off this fatty acid so that can happen, that's going to happen basically right here, and that's going to cost 1 ATP. So if you're going to determine how much ATP is made from this fatty acid or this triglyceride, we're going to calculate how much ATP you make from 1 tail, and that's going to include the cost of 1 ATP for snipping this off. We're going to multiply that times 3 because there's 3 fatty acids and they're all the same length, and then we're going to add the 20 ATP you get from glycerol. So keep in mind, when oxygen is available with glucose, you end up making 38 ATP, right? And you have to have oxygen available for beta oxidation because these acetates are run through Krebs cycle and then electron transport system. So what this is here is this is your fatty acid tail. Let me go back, I forgot to point this out. This is how fatty acids are written. So this is your carbonyl group right here. This is the terminal carbon with 3 hydrogens attached to it, because carbon will only be stable if it's formed 4 bonds. So this terminal carbon right here has 1 bond to the carbon here, so that means it's going to have 3 hydrogens. So CH3 is this carbon. CH214, this is telling you how many acetates, how many carbons there are in the tail that are available for beta oxidation. And then you have your carboxyl group on this end. Okay, so CH3 is the terminal end. The carboxyl end is over here where it was attached to glycerol. And these are the carbons that get chopped up into those 2 carbon subunits. So if you have 14 carbons that are available, that means you're going to make 7 acetates. So this is 14 carbons, and they're grouped together as 2 carbons. So 2, 4, 6, 8, 10, 12, 14. So if you're doing a calculation, you would look at what the formula is. You would know from looking at this that you're going to have 7 acetates, because the number of acetates is your number of carbons divided by 2. So 7 acetates. And then I have the bonds here between them to remind you that for every one of these bonds that's broken between these 2 carbon subunits, you're going to reduce 1 NADH plus H plus and 1 FADH2. So if you group them in pairs, because this is what's going to get chopped off as 2 carbons at a time, you're going to have 7 acetates, and the number of bonds that will be broken is your number of acetates minus 1. So here's your 7 acetates. That's your 14 individual carbons in the fatty acid tail that are available for beta oxidation. And then here are your bonds. Here's 1, 2, 3, 4, 5, 6. So every time one of these is snipped off, here's where the bond is broken. So again, every time you snip off this, you get this acetate, coenzyme A attaches to it, so now you have acetyl-CoA. You break off the bond here, this is going to be another acetyl-CoA. You break off the bond here, 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 and here. So for every one of these bonds that you break, you are going to reduce 1 NADH plus H plus and 1 FADH2. For every one of these acetates that combines with coenzyme A, that's 1 turn of the Krebs cycle. So you have 7 acetates. Now, there's a couple of different ways you can calculate this. You can break them down into the individual products. So 1 ATP, 3 NADH plus H plus, 1 FADH2, that's for 1 turn of the Krebs. You multiply all of these times 7, and then that gives you 7 ATP directly. 7 times 3 is 21 NADH plus H plus, and you would multiply it times 3 because every time electrons are donated to the electron transport system by NADH plus H plus, you make 3 ATP. So 21 times 3 gives you 63 ATP. For every turn of the Krebs, you make 1 FADH, 1 FADH2. 7 acetates is 7 FADH2. Multiply that times 2 ATP, and that gives you 14 more ATP. You can also do this quicker because we already determined that you're going to make 12 ATP for every turn of the Krebs. So a shortcut to doing this would be if you were to multiply 7 times 12 here. And then that should give you the same number. That gives you 84 ATP. Which are from your acetates here. So this should add up to 84. And then for your 6 bonds, remember you're going to reduce 1 NADH plus H plus and 1 FADH2. So that means you have 6 bonds, 6 NADH plus H plus, 6 bonds is 6 FADH2, 6 times 3 is 18, 6 times 2 is 12. So you're going to make 30 more ATP here. Okay, so now we've calculated how much ATP you make from one fatty acid tail. Now you have to take into account that to snip off this tail so that beta oxidation could occur, it's going to cost you 1 ATP, which is this right here. So what this is this is adding up all of the individual ATP that were made if you were to break down the ATP production by the individual components. So you add up all the ATP that's made. This is from substrate level phosphorylation. This is from your NADH plus H plus. This is from your FADH2. This is all from your Krebs cycle. These here are from the bonds that are broken, and this would be the ATP from NADH plus H plus and FADH2. So you add up all of that ATP. Again, this is for one fatty acid. You have to subtract 1 because that's the cost of initiating beta oxidation. So what that means is if you go back here, the bond has to be broken to snip off this fatty acid so that beta oxidation can occur. This bond right here gets broken. So that's going to cost you 1 ATP. So you calculate how much ATP you get for the whole fatty acid, subtract your 1 ATP, and that gives you the total amount of ATP that you get from one fatty acid. Now, for a homotriglyceride, all 3 fatty acids are the same. So you would multiply this times 3, right? 3 fatty acid tails, that's going to give you 339 ATP. And then you add in the 20 ATP that you get from glycerol. So that means from this triglyceride, which is kind of on the smaller end of a triglyceride, you make 359 ATP. Do you remember how many ATP you make from glucose? You make 38. So for a relatively small fatty acid, you can make a lot more ATP. So it makes sense why your liver and skeletal muscle particularly will preferentially metabolize fatty acids over glucose, because you get a lot more ATP out of those fatty acids. So what you want to be able to do is you want to be able to do this calculation, because you will get tested on it. You want to know what you get for 1 turn of the Krebs. You want to know that the fatty acids get broken down into individual acetyl-CoAs, and every one of those is going to represent 1 turn of the Krebs cycle. For each bond between the acetates, you're going to reduce 1 NADH plus H plus and 1 FADH2. And you need to remember that glycerol gets converted to 3-PGAL and enters glycolysis at that point, and that you make 20 ATP from it. Also remember your cost of beta oxidation, which is the cost of snipping off this fatty acid so that beta oxidation can occur. So again, this is one way to add up the ATP. The shortcut that I gave you would be for every acetate, or for every turn of the Krebs cycle, you're going to make 12 ATP. So you can multiply the number of acetates times 12. For each bond, you can do the same thing. For each bond, you're going to make 3 ATP here and 2 here. So for each bond, you make 5 ATP. So you can multiply the number of bonds times 5. That would give you your total ATP for that fatty acid. Subtract the 1 ATP for the cost of beta oxidation, and that'll give you your total ATP from 1 fatty acid. In all of the examples that I would give you an example of a homotriglyceride. So you could simply multiply this times 3. You could, let me go back a couple of slides. You could do the calculation if you had a typical triglyceride that had separate fatty acids. But there's no point in that. You should be able to just understand the concept. And again, just to make sure you do, you need to be able to do this calculation. So you have your total number of ATP from 1 fatty acid, multiply it times 3, because you have 3 fatty acid tails. That's your total number of ATP from a fatty acid. You add in the 20 ATP you get from glycerol, and this is how much ATP you can get from one relatively small triglyceride. So again, when your body is going to metabolize a lot of lipids, you make a lot of acetyl-CoA. So some of those can be used to make ketones or ketone bodies. So ketosis is when the liver forms these ketones from fatty acids or other tissues as fuel source instead of glucose when glucose levels are low. So your body can go into ketosis. Now, if you're doing the keto diet, that's the goal, is to get your body into ketosis, where you are basically burning all of your fatty acids instead of your glucose. So your ketogenic diet that's high in fat, high protein, very low carbohydrate. So you want, this is the goal for the keto diet, is to get your body into ketosis. Now, this isn't something that your body would normally do and sustain. It can be done temporarily if you have, you know, some situation where, let's say you're fasting unintentionally for whatever reason, or you have a really intense workout. So those are some things that can cause your body to go into ketosis naturally, but normally you wouldn't do that for a prolonged period of time under normal conditions. But ketones, these ketones that are produced, can be used by your brain, your heart, muscle, and your kidney. So it takes these subunits and converts them into ketones, which then can be used as a fuel source in these other tissues. There is also what's called diabetic ketoacidosis. So ketosis is basically you're producing these keto acids. Keto acids are acids, right? Pyruvate is a keto acid. We've already talked about the fact that acids can't hang out inside of the cell because they would alter the pH. So in ketosis, when you are eating all of these high fat, high protein things in your diet, you're basically producing a lot of acid. And so that means your body has to ramp up how it deals with acid. Because again, in a healthy individual, that acid isn't going to be built up. Your body's going to deal with it. It's going to match the acid production with bicarbonate production, and it'll increase filtration by your kidneys to get rid of it so that your body fluids don't become acidic. In diabetic ketoacidosis, though, remember, for someone who's diabetic, their cells are not taking in glucose out of their blood. So if cells can't take in glucose, they don't have that available as a fuel source, but what they end up metabolizing are fatty acids. And because this is happening throughout their entire body, they are producing more acid than their liver and their kidneys can keep up with. And so they do end up with their tissues becoming acidic. And this is a very dangerous situation. Typically, someone who's gone into ketoacidosis from diabetes, they typically end up on dialysis because they need help clearing that acid from their body. So this is not a normal thing that happens in physiology. For a short period of time, this is totally normal and can happen all the time. But typically, if you're eating a balanced diet and you don't have any pathologies, this won't be sustained for any length of time. And then I wanted to show you this because this is a summary of all of the metabolic pathways that happen inside of a cell. So here is glycolysis, here is Krebs cycle. But just notice all of the lines coming into and out of all of these pathways. So when we talk about metabolic intermediates and their importance in terms of being able to enter and exit other metabolic pathways, and when I also say you produce all of this acetyl-CoA, and what happens to it depends on the needs of the cell. So here's acetyl-CoA right here. So if the cell needs to make ATP, it'll run it through Krebs electron transport system. But there's a lot of things that cells need to do to maintain homeostasis. And so it may need to do any one of these things rather than make ATP. So if you take biochemistry, you'll get into more of these reactions. But right now we're just focusing on these. So that is the end of our discussions of cellular metabolism. In the next lecture, we will start talking about cellular communication.